My name is Dr.S.Radha, Assistant Professor from Department of Microbiology, the Standard Fireworks Rajaratnam College of Sivagasi. So, today I am going to discuss about a subject or a topic of forensic science. So, this topic what I have going to explain is of AFLP, that is amplified fragment length polymorphism. I am going to discuss about how the DNA is extracted for this process and what is the basic principle behind this and how following the method step by step this is done and for what reason we are doing this, that is application of this technique. So, amplified fragment length polymorphism is the technique, a PCR based technique that is used as selective primer and that focus on the arbitrary sequence. So, here we are fragmenting the DNA sequence and we are amplifying that and thereby we are detecting the polymorphism. And so, this technique involves the steps. So, starting with first the restriction digestion of the genomic DNA of the source what we are going to take for the forensic study. The DNA extractor is fragmented with restriction enzymes and we are ligating that with the adapter sequence. The adapter sequence will be attached or pasted to the end of the fragment and then we are going for a pre-selective PCR technique where the primer will be selecting the fragment and that will be amplified by using a PCR technique and we will be visualizing the result by electrophoresis. Finally, on gel we will be scoring and interpreting the data. So, here I have shown the diagrammatically how we are doing this process. So, first it starts with the DNA extraction and we are fragmenting. The fragment will be of different size we will be obtaining starting from that is 50 to 500. So, based upon the restriction site location the fragment will be available and that will be ligated with the adapter sequence. Adapter is nothing but the adapter sequence is a synthetic sequence that is having a restriction site and that will be pasted to the blend end and this will be followed by adding the primer. That primer will be specific for the arbitrary sequence and that will be designing for forensic science and satellite based upon the satellite sequence and that will be amplifying the that is target gene. And finally, the PCR product will be run on the gel. The gel will be getting the various band pattern based upon the result of AFLP. So, here I have focused the primer what we are using and how this ligation we are doing. So, ligation of the restriction fragment will be done to with the adapter sequence and this primer will be designed based upon the arbitrary nucleotide sequence available that is nothing but the satellite sequence that will be complementary to the arbitrary nucleotide. And this is a technique that is used to see the polymorphism. For that the primer is considered to be very important. So, here in the diagram I have shown how in the first step the restriction enzymes are used to cleave the DNA sequence. DNA is fragmented and next the restriction we are adding the adapter and adapter after ligating we are going for the pre-selective amplification PCR method and finally we are doing a PCR. Under visualization we are running the sample or obtain the product in the gel. Then we are using usually the denaturation polyacromide gel and by the principle of autoradiography or if you are using a fluorescent dye we are using fluorescent technique to observe the result and that will be analyzed. So, what is the advantage of this technique is means it is highly reproducible, means we can repeatedly we can do and it will be more, that is we perfectly we will be getting the result. It is having a high resolution, even a small amount of sample it is enough and we can directly use a genomic DNA. The fragment ranges from 50 to 100 that is times that is we are getting and it is considered to be, it does not require any sequence details. So, in unknown sequence we can do it. Then why we are doing this, that is the application of this technique that is considered to be very important. So, it is used for in various polymorphism studies where genomic analysis is done to compare the cultivar or compare the related species and the genetic variation among the strain and related to plant, animal in any microorganism, fungi, bacteria we can study. And more specifically in forensic science it is used to observe the criminal and paternity test where we are choosing the particular person with the closest match. And not only that, it has a great application in popular genetics, that is population genetics, under which we can see the differences among the population or races we can check. That is all about this description. Thank you.